Hi everyone. Today we are going to solve the exercises of chapter 1, page 30 31. We are going to start with the first exercise, exercise 1. Determine whether each statement is a true or false and correct the false statement. We have to answer by true or false, and then if the statement is false, you have to correct it. 1. A plant develops on a strictly mineral medium in the presence of light. Is it a true or false? I'm going to ask you. Does is any plant capable of living on a strictly mineral medium in order to survive? Of course, not any plant. The green plants, they can develop on a strictly mineral medium. What do I mean by mineral medium? Medium rich in water and mineral salts in the presence of light. So by this, the green plant will be able to make what? Photosynthesis. So the answer is false. A green plant develops on a strictly mineral medium in the presence of light. You have to rewrite all the statements. Number 2, a green plant can synthesize. What do you mean by synthesize? Produce organic materials, of course I mean by organic material starch, from mineral substances by photosynthesis. This statement, it seems to you that it's a true, and it is true, but here when we say photosynthesis, we have to be more precise, and we have to say in the presence of light. So the green plant can make starch from the mineral substances by photosynthesis in the presence of light. Number 3. A green plant in the presence of light only absorbs oxygen and releases carbon dioxide. In the presence of light only absorbs oxygen and releases carbon dioxide. In other words, they are telling you that the green plant in light only respire. No, it doesn't only respire. It also what makes photosynthesis. So we say a green plant in the presence of light absorbs CO2 and releases O2. Number 4. A green plant respires only in darkness. Only it respires in darkness. Of course, no. The green plant, like us, respire day and night. The last part of number 1. A green plant exposed to light needs only water and minerals for its growth. It needs only water and minerals. What else does it need? It needs an important factor, which is CO2 for its growth. Number 2. Choose the correct answers. You might have one answer true or more than one true answer. Let's read. By photosynthesis, a green plant absorbs carbon dioxide? Yes, true. Absorbs oxygen? No. Synthesizes starch or produces starch? Yes, true. Releases oxygen? Also true. So A, C, and D. Part 2. A green plant exposed to light can develop, a plant exposed to light can develop in a mineral medium. As I said before, not any plant can develop in a mineral medium. The green plants are able to develop in mineral medium because they are able to make photosynthesis in the presence of light. So the first one, it is false. B. Can develop in an organic medium? Yes, any plant can develop in an organic medium, because when I say organic medium, I mean by this medium rich in starch, as if I'm telling you that this plant, it is living in a medium where its food is available, so it can survive normally. C. Can develop in a mineral medium if it is chlorophyllc? True. Only if it is green, it can develop in a mineral medium in order to make photosynthesis. Cannot develop except in a mineral medium if it is chlorophyllc. They are telling you cannot develop except in a mineral medium if it is chlorophyllc. No, yes, the the chlorophyllc plant can develop in mineral and organic medium. Part 3. A green plant undergoes photosynthesis when it is placed in air. Yes, this will allow it to absorb carbon dioxide. Enclosed under a jar in darkness, of course no. Here light will be absent. Exposed to light, yes. Exposed to light in an atmosphere deprived of carbon dioxide. What do you mean by deprived? It means without. Can the plant make photosynthesis without CO2? Of course no, so it is wrong. So part 3, A and C. With respect to exercise 3, determine the term that doesn't belong to each set of terms. We've determined the term and I've made a statement for the other words. Okay, we've put them in a statement. You have chloroplasts, you have starch, you have mineral medium, you have non-chlorophyll plant. Which one is the intruder? What do I mean by intruder? As if I'm telling you remove the one that doesn't belong to this set of words. Here we can say that the non-chlorophyll plant. We can say photosynthesis takes place in the chloroplast by which the mineral materials are used to synthesize what? Starch. Part 2. Photosynthesis, darkness, oxygen, carbon dioxide. We say here we have darkness because photosynthesis takes place what? In light by absorbing carbon dioxide. Part 3, green leaf, heterotroph. The green leaf, the heterotroph, they depend on the autotroph as I mentioned before. We have mineral substances, we have organic material, we have chlorophyll, we have autotroph. Which one doesn't belong to all these words? We have the heterotroph. Why? Because the green plants, they take mineral substances to synthesize organic material by synthesis, okay, which this refers to what? To the autotrophs. Number 4, it's important. Listen please. A student testing the hypothesis that carbon dioxide is an indispensable factor for photosynthesis. So student, he is asking himself if is carbon dioxide important for photosynthesis? What do you mean by indispensable? It means essential or important, okay? He's asking himself, is carbon dioxide important for photosynthesis? He wants to test his hypothesis, whether it's true or false. So he performed an experiment. Let's see what was the experiment of such a student. Places a pelargonium plant under a jar where air is enriched with carbon dioxide, and he covers the jar with an opaque cover. So he got a pelargonium plant, he placed it in a jar containing air with carbon dioxide, and he covers the jar with an opaque cover. What do I mean by opaque cover? Opaque cover, it is the cover that doesn't allow the entry of light. So here the student, he's working on which factor do you think in such experiment? On light or on CO2? He's working on, it appears that he's working on a light because he placed an opaque cover. Let's check whether his experiment is true. 10 hours later, after 10 hours, when the leaves are treated in hot alcohol and iodine water, after 10 hours he applied iodine tests. They show yellow coloration. They give a yellow coloration. What does this coloration indicate? Of course, I'm going to remind you that with iodine test, if we get a dark blue color, this means that there is starch. And if we get a yellow coloration, this means that there is no starch. So the yellow coloration, it indicates the absence of starch. We conclude that the photosynthesis doesn't take place. What conclusion can be drawn? We can conclude that light is necessary for starch synthesis. Part B. The student doesn't expect this result. The student, he doesn't expect this result. Help him find the error that he has committed while experimenting. What do you think? What was the error that he have made while making the experiment? As I mentioned before, that when you want to make an experiment, okay, you have to put one variable in order to be able to conclude the importance of such variable. And here the student, He worked on two variables. His hypothesis was about carbon dioxide, and his experiment was about another variable, which is light. And this was clear by placing an opaque cover. So his work was wrong. We have to tell this student that while experimenting, you have to act on one variable and all the other factors should be kept constant. Okay, so in the experimental procedure, the students work on two factors: light by placing an opaque cover, and CO2 when they were testing the hypothesis, asking themselves about the hypothesis. So we can tell him that to study the importance of one variable, other factors should be kept constant. So his experiment is dealing with two variables, and this is not true. Let's move to exercise 5. What do we have in such exercise? Two setups A and B. Look at A, look and B, at B. Containing a pelargonium plant, a green plant, are covered by a plastic bag. Both of them, they are covered by a plastic bag. Setup A has a small vessel containing sodium bicarbonate. So A, it contains a small vessel containing sodium bicarbonate. If I ask you to remind me about the role of sodium bicarbonate, if you remember, sodium bicarbonate, it releases carbon dioxide. In other words, it provides such a plant with carbon dioxide. While setup B has its air previously traversed in a lime water vessel, it absorbs carbon dioxide. The two setups are exposed to light for 10 hours. So all the conditions, they are available. They are exposed to light, they are green, and they are A is supplied with carbon dioxide, while B, if you notice, there is no carbon dioxide. The question is, how can the presence of starch in the leaves obtained from both setups A and B be identified? If I want from you to tell me how can we test the presence of starch, of course, you have to say by iodine test. What would be the result in each case? In leaf A, since all the conditions for photosynthesis are available, like green color, chlorophyll, water, mineral salts, light, CO2 from sodium bicarbonate, so the iodine test gonna give a dark blue color since all the conditions are available. So this plant will make starch. And with respect to the plant B, okay, this plant will gonna give with iodine test a brown orange color because it doesn't contain carbon dioxide. So there is a missing factor important for photosynthesis, that is carbon dioxide. So the color will be brown orange color. What can be deduced here? Which factor we are studying? We're studying the importance of carbon dioxide. We can say sodium bicarbonate releases carbon dioxide, which is an important factor or indispensable factor for photosynthesis. Now let's move to exercise 6. It's important. Okay. The graph below represents the variation in the volume of oxygen released by green algae exposed to light as a function of luminous intensity. We are checking how much the volume of oxygen is being released by this green plant with respect to the light, to the luminous intensity, which is expressed in lux. Okay, what do we have? Look at the graph. As the intensity of light is increasing, if you notice till 30, the volume of O2, more volume of O2 is released by such a plant. In other words, as the volume of O2 increases, you can tell me there is more photosynthesis. But above 30, if you notice, yes, above 30 lux, the volume of O2 decreases sharply. What does it mean? I'm going to ask you, high intensity of light, is it good for the green plant to make photosynthesis, or it is not good? As it seems from here that the high intensity of light is not good for the green plants. Because why? Because the high intensity of light is going to destroy the chlorophyll, the chloroplasts, and mainly the chlorophyll, and by this the green plant will not be able to make photosynthesis. You are asked to interpret such a graph. I will remind you about the interpret of a graph. On the x-axis we have variable, on the y-axis we have the result, and when we want to interpret, you have to relate the variable with respect to the result. Okay, what was the result at a certain with respect to the variable that is given. Okay, so how do we interpret? We say when the luminous intensity is nil, the volume of O2 released by the algae is 0 AU. It is nil. We have to put connecting words. Now we move from 0 to what? To 30, because it kept increasing. Then we say whereas when the luminous intensity increases from 0 lux to 30 lux, the volume of O2 released by the green algae increases from 0 to a maximum value equal to 60 AU. Look at the maximum value by the ruler, make it horizontal or parallel to the x-axis, you will get the value. It's equal to 60 AU. Whereas when the luminous intensity increases from 30 to approximately 33 lux, the volume of O2 released by the algae decreases from 60 to 0 AU. So what can you conclude as a relation? So what? So the volume of O2 released by the algae, is directly proportional to the luminous intensity to a certain limit, beyond which it decreases what? Rapidly. So the plant is going to release more O2, okay, when light is available, but to a certain limit. If the frequency of light gonna increase too much, this plant will not be able to make photosynthesis, okay, and not going to release more O2. Now let's move to exercise 7. What do we have in exercise 7? The graph below represents the variation of the volume of oxygen produced by an Elodea twig placed in light as function of the amount of carbon dioxide present in water. Now we are checking the importance of carbon dioxide with respect to photosynthesis and mainly with respect to the volume of oxygen released. As I told you before, that you can know that there is photosynthesis if they told you the volume of O2 released. If it is more released, it means that there is more photosynthesis, okay, and by the amount of starch produced. Okay, so let's check the how photosynthesis is being affected by the concentration of carbon dioxide. As you notice in such a graph, many students ask me that on the y-axis there is no values. Okay, it is normal. If there is no value, you should not put any extra value from your knowledge. It's wrong. You have to interpret or analyze such a graph as it is. So when you want to use the values, since on the y-axis there is no value, you can say it increases to the maximum or to a certain limit and then it remains constant, for example. Okay, so the first question you are asked to interpret the graph. Let's make interpret. We say when the concentration of CO2 is 0, the volume of O2 released by the green plant is 0. Whereas when the concentration of CO2 increases from 0 to 0.3, look how it is increasing. The volume of O2 released by the green algae increases from 0 to a maximum value. So what? So the volume of O2 released by the green algae is directly proportional to the concentration of what? Of CO2, where its abundance enhances the release of O2 by the green algae. So the availability of carbon dioxide gonna make photosynthesis to take place more and the green plant will release more oxygen. Then in part 2, what do we have? The value 0.03% of carbon dioxide represents its amount present in air. How can the atmosphere of a greenhouse be enriched with carbon dioxide? If you have a greenhouse and you want to enrich the environment with carbon dioxide, how can you enrich it? Simply by burning dry branches. These dry branches, due to burning, they are going to release carbon dioxide, and by this way, this green environment, this greenhouse gonna be enriched with carbon dioxide. Part 3. Why do we say that green spaces are the lungs of the cities? Why the green spaces, they are the lungs of the cities? Why in cities like Beirut, why green plants or green areas are important for such city? You know that in the cities, the cars and the factories, they produce large quantity of CO2, right? That is used by the green plants to produce what? O2, thus making the atmosphere healthy. So more areas of green plants, okay, this will make these green plants to release more oxygen by absorbing the high quantity of CO2 that is being released by the cars and the factories, okay, and by this more oxygen will be available in the atmosphere, and this will make it more healthy. As a conclusion, photosynthesis increases as the concentration of CO2 what increases. Number 8. Most leaves are placed under the following conditions. We get green leaves, sorry, and they were placed under different conditions. First case, open air and light. Let us discuss together. If the green leaves are placed under open air and light, will they make photosynthesis? Of course, yes. Air deprived of carbon dioxide and light, air that doesn't contain carbon dioxide and light, here there is one missing factor for photosynthesis, so this plant will not make photosynthesis. Open air and darkness, here is the same, there is one missing factor which is light, so this plant will not make photosynthesis. The first question: draw a labeled diagram of a moss leaf placed in a drop of water under the microscope. When they tell you under the microscope, they are asking you to draw what? Of course, you know that under the microscope you are able to see the cells, right? As if he is telling you to draw the cell of such moss leaf. And since this moss leaf, it is a green leaf, it's an algae, a green leaf, so it contains what? Chloroplasts. So like any cell, any chlorophyll cell, it possesses cell wall, cell membrane, cytoplasm, nucleus, vacuole, and the chloroplasts. Then, how would the cells of the leaves in A, B, and C appear if each is placed in a drop of iodine water? They told you what would be the result of iodine test in the presence of, in each of the three cases. In A, since all the conditions are available, iodine gonna give a dark blue color. In B, it is going to give a brown orange color since there is no CO2, and in C it will give a brown orange color since there is no light. Now let's move to exercise 9. 9 is very, very important. It includes a new idea, and please listen very well in order to understand. Exposed to a radiation spectrum, as I said, as I want you to know that radiation spectrum, it is the, you know that each color, it has a specific wavelength, and the colors of the rainbow, they form a spectrum, right? And each one corresponds to a specific wavelength. So they got a radiation spectrum. Look at it. Which colors? Violet, green, and red. They got a Spirogyra, a green algae or a green plant. They are telling you that we've got a green algae or a green plant, and it is exposed to radiation spectrum, as if they are telling you that this green plant, it is being exposed to different colors of light. I've got the violet color of light, a green color of light, and a red color of light. With a colony of aerobic bacteria, Look please, this is the green plant, the Spirogyra filament. It looks like a spiral. Yes, from its name, Spirogyra filament. It's a green plant, green algae. And this green algae, it's exposed to red color of light, green color of light, and violet color. Is what more does it possess? They told you with a colony of aerobic bacteria. Where are the bacteria in such diagram? You see the black dashes? Yes, the black dashes, they represent something called aerobic bacteria. So they are the aerobic bacteria. And if you see that we have different concentration of aerobic bacteria depending on the light. In the red light, we have the highest concentration of such aerobic bacteria. In the green light, we have the least amount. And in the violet color, we have also available amount, right? Good amount. So, what is the explanation of such exercise? What I want you to know. First of all, I want you to know the meaning of aerobic bacteria. If I ask you later or in any other exercise, what do I mean by aerobic? Aerobic bacteria, it means bacteria that needs oxygen. So what do I mean by aerobic bacteria? Bacteria that needs oxygen. And this bacteria, you see that it is spread in different proportion. In which light it is the highest amount, highest frequency? In the red light. Look how many black dashes do we have, right? In the red, then in the violet, and the least amount of such bacteria were concentrated in the green light. Why? Why do you think? You have to explain. The green plant, this green algae, it is making photosynthesis in different proportion, okay? In the red light, it is the highest, then in the violet, and the least amount was done in the green light, okay? So, why? Uh, how do you know that photosynthesis is taking place more in the red radiation? So, what do we have here? They ask you in number one, because the questions, they are the explanation for the exercise. Why do the bacteria approach the algae? Why do these bacteria, they approach the algae? They come near the algae. As I said, that these bacteria, they are aerobic. And aerobic, it means that they need oxygen. You can explain for me in such a way. And it approaches the algae because this algae is green and it is capable to make photosynthesis. And by making photosynthesis, such a green plant, green algae, is going to release oxygen, which is needed by such bacteria to survive. So the bacteria comes near or approaches the algae in order to take the oxygen that is released by photosynthesis. Okay, so we say the bacteria approaches the green plant or the green algae to obtain oxygen that is released by photosynthesis. They ask you, would the bacteria behave in the same way in darkness? This bacteria, will it approach the green plant in darkness? No. Why? Because in darkness, such a green plant doesn't make photosynthesis because there is no light. And since there is no photosynthesis, so it will not release O2. And since there is no O2, so the bacteria will not approach the green plant. Okay? Number three is very important, and a new idea is being introduced here. What can be deduced from the different distribution of the bacteria? As you see in the document, that the bacteria is distributed in different proportion according to light, right? And as I said, it is distributed in the red color the most and the least in the green color, and the violet color it is almost equal to the red one. Why? Listen please. You have to know that the plants, they react differently to different colors of light. So if you get a plant and you place it under red light or under green light or under violet color, light, this plant is not going to respond in the same manner under these three lights. They will react differently. Why? Because as you see, the green light, it is the least effective for the plants. The green plants, they are going to react the least in the least manner, okay, under the green light. Why? Because the green plants, from their name, green plants or chlorophyll plants, they are green, and the green light is also green. So so when they are exposed to green light, green and green gonna reflect each other, and this will not allow such a plants to absorb enough energy from the light. So photosynthesis will be the least. Okay, so the green plants, they possess a green pigment called the chlorophyll, which provides them the green color. And by exposing such plants to a green light, this will not allow them to absorb enough energy. Okay, so photosynthesis will take place, but under a very low amount or frequency or concentration. Under which light it is taking place the most, as you see in the figure, under the red color. How do I know? By the concentration of the bacteria. We said that these bacteria, they are coming near the green plant because it is making photosynthesis, and they possess a big number under the effect of red light. So the red light, under the effect of red light, the plant will be able to absorb a high amount of energy from the light. So photosynthesis takes place to the maximum under the effect of red light, then under the effect of violet light, but it doesn't take place, it takes place to a least extent under the effect of a green light. So from here we have to know that the plant absorbs mostly blue and red light, okay, and they rarely absorb green because the green light, it is being reflected by this plant because it is also a green plant. So green color and the green color gonna be reflected by the green plant, while the red color and the violet color, they are going to be absorbed by the plant, and photosynthesis will take place to the maximum. As a conclude, let me answer. A great number of bacteria direct themselves toward the red radiation or the red light, which indicates the production of a great number of O2, as if we are saying photosynthesis is taking place to the maximum. Thus with red radiation is maximal، كأنه عم نقول photosynthesis is maximum with red radiation. Photosynthesis is nil with the green radiation, and with violet radiation it is nearly the same as in red radiation, and we can say it is also maximal or maximum. Okay, any question, please ask me, my lovely students. Thank you.